Manipulation of plasmid DNA with alkaline lysis. Label 2 centrifuge tubes. Add broth in both the centrifuge tubes. Centrifuge both the tubes. Keep pellet, discard supernatant. Add solution 1 and vortex it up to pellet get mixed. Add 200 ml solution 2 and 150 ml solution 3 simultaneously. Keep the tubes on ice for 5 minutes. Centrifuge the tubes. Transfer the supernatant in new tubes. Add equal amount of phenol chloroform in both the tubes. Centrifuge the tube. Ring observed after centrifugation. Transfer the aqueous phase in new centrifuge tube. Add isopropanol, double amount of aqueous phase. Centrifuge the tubes. The pellet. Centrifuge for 2 minutes. Keep the pellet. Keep the tubes open. Add nucleus-free water. Add the plasmid sample and gel loading dye. Gel filtration unit. Load the sample on agar gel for electrophoresis. Keep it for 2 hours. Observe the bands. Reagents used and their concentrations. Restriction digestion of PUC19 plasmid. Principle. Procedure. Take 1 microcentrifuge tube. Add the following in microcentrifuge tube. Centrifuge the sample for few seconds. Incubate the sample for 1 hour in water bath. Add 4 ml gel loading buffer in 20 ml plasmid sample in new tube. Load the sample on agar gel for electrophoresis. Results. Reagents used and their concentrations. Double digestion of PUC19 plasmid. Principle. Procedure. Label and add the following in a microcentrifuge tube on ice. Centrifuge the sample for few seconds. Keep the sample for incubation for 90 minutes. Add gel loading dye in a sample. Load the sample on agar gel for electrophoresis. After double digestion of PUC19, two observed. Agents used and their concentrations. DNA extraction. Procedure. Measure the weight. Cut the DNA band present on a agar gel with sterile blade and transfer it into same tube. Add 0.916 ml sodium iodide. Incubate the vial for 10 minutes at 55 degrees Celsius to solubilize agarose. Add 1.5 ml silica beads into the vial and keep it for 10 minutes at room temperature. Centrifuge the tubes. Discard the supernatant and keep the pellet. Add 200 ml wash buffer in a vial containing pellet. Vortex the vial for few seconds. Centrifuge. Incubate the sample at 55 degrees Celsius in dry water bath. Add 20 ml nucleus free water. Again keep the sample in dry water bath. Add 10 ml purified DNA in 2990 ml distilled water. Take absorbance at 3 different wavelengths. Reagents used and their concentrations. BlueWhite screening. Transfer the culture into 2 tubes equally. Principle. Centrifuge the tubes at 4000 rpm. Resuspend the pellet by adding 5 ml of ice cold CaCl2 in each tube. Again centrifuge at 4000 rpm. Resuspend the pellet with CaCl2 and 800 ml glycerol in each tube. Collect 100 ml of culture into 2 1.5 ml vials each. Add 5 ml ligated sample and 5 ml control into separate 100 ml competent cells. Keep the tubes in incubator at 42 degrees Celsius for 19 minutes. Keep the tubes immediately on ice. Add 800 ml of LB media into both the tubes. Keep the tubes at 37 degrees Celsius for 45 minutes in shaking incubator. Give pulse with table centrifuge. Spread 100 ml of transform and control cells on X-gal IPTG Loria agar plates containing ampicillin. Incubate both the plates overnight. Results: transformation of bacteria to express GFP. Principle: measure 1.5 gram of agar powder. Add 1.5 gram of agar powder in 100 ml of distilled water. Measure 12.5 gram of LB for 500 ml of distilled water. Stir it well. Add 400 ml of LB to 6 gram of agar. Add 100 ml of LB to 1.5 gram of agar. Autoclave it. Pour media into Petri plates and allow it for solidify. Add 11 ml of water. Add 2 ml of vector. Add 4 ml of insert DNA. Add 2 ml of ligation buffer. Add 1 ml T4 DNA ligase. Vortex it. The vials were kept in a refrigerator at 4 degrees Celsius overnight. It was kept on shaker at 37 degrees Celsius overnight. Culture is transferred equally into 2 tubes. Centrifuge at 6000 rpm for 10 minutes at 4 degrees Celsius. Discard the supernatant completely. 1 ml of ice cold calcium chloride is added. Add resuspension 8 ml of ice cold calcium chloride. Tubes are kept on ice for 20 minutes. Centrifuge it for 20 minutes at 3500 rpm. Discard the supernatant. Again 850 ml of calcium chloride added. Allocated 100 ml of resuspension into 8 tubes. Heat inactivate the ligated sample at 65 degrees Celsius. Add 10 ml ligation sample and 1 ml control DNA in 100 ml of competent cell containing 2 vials respectively. Keep the vials in water bath for 2 minutes. After that, keep the vials immediately on ice for 5 minutes. Add 0.5 ml LB broth in both the vials. Keep the vials in shaking incubator. Label 2 LB plus ampicillin plate as ligated sample and control and spread the respective sample on it aseptically. Place the plates in incubator for 24 hours. Results: reagents used and their concentrations. Thank you.